Thanks so much. Thanks for the invitation. It's a real honor to be in this workshop. This morning was just such a tour de force in these invertebrate systems and seeing how you're really starting to get answers, not just questions, but actual answers. I wish I could say the same. What we've got here is some data, but it's nothing like the level of understanding we've heard this morning. But hopefully, maybe it's going, going in the same direction. This is mainly published work in this paper here. I'm going to try and get to some unpublished stuff at the end if there's time. It's work that was driven by Stéphane Boujon, a vast team of people in the Victoria Reddy and Matteo Carandini and I run at UCL, and it was 7 years of work. The cortex, as has been known for over 100 years, contains many different types of neurons, and originally they were classified by their anatomy, as shown in this lovely Lorente Deneau picture. And one of the questions of mammalian neuroscience, at least we now do have an answer to, is what are these cell types? So the morphology gives you a lot of clues, but in the last few years, we've actually got answer to the question definitively of what are the cortical cell types from transcriptomics. And I think everyone, I hope, knows a bit about transcriptomics by now. We saw a few talks related to it in the main talk, but to go into a bit more in the main session, to go into a bit more detail, transcriptomics characterizes cell groups by their combinatorial expression of multiple genes. So no one gene is enough to identify a cell type, but the whole ensemble of genes in the genome, which ones are expressed in a given cell, is really the best way we've got now to classify different cell types. There are two techniques of transcriptomics, the main one being single cell transcriptomics, which is when a piece of tissue is digested, the axons and dendrines sheared off the cells and they're turned into spheres, and then you count the expression of all genes in every cell. This is the best technique for getting data, but it loses information about where the cell was in the tissue. The other method that's more of a new approach, in situ transcriptomics, you apply it to a tissue slice. It keeps the location of a cell, but it only measures selected genes, a few hundred genes. But that's still enough to classify the cells you look at. All right, so let's look at some single cell transcriptomics first. The data you get is a matrix of expression levels, number of cells by number of genes. You can apply cluster analysis to it if you want to define cell types, but cluster analysis by its definition gives you clusters. The actual cell types may lie on a continuum. So let's look at some data from this paper here from the Allen Institute, Tasic et al. 2018, which is what really is the best definition of cell types in V1. So this is a UMAP analysis. It's a way of putting the cells in 2 dimensions such that cells that are close together in this 2-dimensional space are close together in the original space of gene expression. And this is applied to the inhibitory neurons of primary visual cortex. They're divided into 2 main groups according to their developmental origins. And you can see this very complex picture. You might have heard a few years ago people saying there are 3 cell types of inhibitory neuron. Obviously, that's absurd when you look at this picture here. You might remember the talk on the SST-NOS1 neurons there here. We've got all these different types of interneurons, and some of them really are discrete clusters. So for example, this cluster here, we're now zooming into this half. This is showing the same points, but with the marker size, expression of VIPR2, it's a VIP receptor. That's only found in this cell type. This is a very discrete cell type. This happens to be chandelier cells, axoaxonic parvalbumin cells. SST-NOS1 is there. There are other discrete cell types. But as well as that, there's continua. So if you look over here, this is a group of neurogliaform cells and other inhibitory neurons. And if you look at it, there's a continuum, there's a full continuum. And the different colors and glyphs tell you different clusters that came out of the cluster analysis, but they're not really discrete clusters other than this one. They're just tiling a continuum. So if we look into it, now we're giving the size of these points by expression of one particular gene, NDNF. And what you can see is a continuum, that the level of MDNF expression goes down in this direction and continuously down in this direction. The cluster analysis draws an arbitrary boundary along this continuum, but it's really a continuum rather than discrete clusters. If you apply cluster analysis, however, you get clusters, but you have to remember the data may or may not be continuous. All right, so that was a bit of background. What I want to talk about is how different is the in vivo activity of these molecular subtypes. So Passage et al. Defines 61 subtypes of V1 inhibitory cells by cluster analysis. And this is an underestimate in the sense that within each of these clusters that they found, there's still statistically significant variation in the amount of RNA expressed. The question is, do these cells all have different functions in vivo, meaning different correlates of what they fire with in relation to sensory stimuli, etc.? If the answer is no, then our job as neuroscientists is easier. Computational neuroscience in the old days had one type of neuron, then it graduated onto having two types of neuron, and maybe 5 or 10 years ago we started to have 4 if you count the 3 so-called inhibitory classes and 1 excitatory. But when it gets to 61 types or 100 types, then things get really difficult. If it was the case that actually you don't need to model all of those types, life is much easier. If, on the other hand, as you've got to worry, it will be that there is a difference in the function of these types, otherwise, why would they have different gene expression? Then at least are there some simple principles that can help us understand what's going on. So the way we approach this is with 2-photon calcium imaging in vivo, then we cut slices of the brain, and then we apply in situ transcriptomics to identify the fine molecular types of the recorded cells. We're basing our cell types on the Passage et al. Classification, but because we're imaging only the superficial layers of cortex and focusing on inhibitory cells, we don't have the full 61 inhibitory subtypes, only about half of them, which live in the superficial layers. And we've divided these subtypes into a 3-layer hierarchy. So the clusters are here. At the top level, what's the Passage called the subclasses are the 5 main types of inhibitory neuron. You've got the famous ones, parvalbumin, somatostatin, and VIP. There's 2 others, LAMP5, which is mainly neurogliaform cells, and SNCG, which are what used to be called CCK cells. Some of these have VIP as well. Anyway, we've got these top 5 classes, and we've got these 30-something clusters in the superficial layers. But what we did is we defined an intermediate level of 11 subtypes, 11 cell types. And these correspond to the ones that we can correspond to classically defined cell classes. So for example, this one here that we saw earlier, PVAL VIPR2, that's chandelier cells. That's a type that contains only 1 subtype. But this type here, PVAL TAP1, contains 5 subtypes, but we don't know what the difference between them is. We just know that they're all basket cells, parvalbumin basket cells. So we've got 11 types. These are the ones that we have some sort of biological interpretation for. We've got the subtypes, which are the variability. We don't know what it means. And then we've got the subclasses, which is the 5 top-level types. And this map here shows you the expression of 73 genes, which is the panel that we used to analyze the cells in the brain slices that we cut. This is what in situ transcriptomics looks like. These are the 73 genes in our panel. And incidentally, 73 is about all you need. We started with 100 and got rid of 25 or so because they weren't helping the classification. So even though the genome is large, around 100 genes is all you need to classify these cells very, very well. So these different symbols and glyphs, colors and glyphs should tell you the different gene codes, and you can see the different cells in different colors because the glyphs are colored so that single cell types tend to have the same colored genes. If we zoom into this region, one of the places we imaged, now you start to see the individual cells, and if we zoom into individual cells, you can see the reads, and just by looking at these, I could tell you that this is somatostatin, COHBP, reelin, that's a Martinotti cell. This one, it's got IV2, it's got LAMP5, that is a IV cell or an MGE-derived neurogliaform cell. When you look at these diagrams for long enough, you'll start to recognize them. But the point is that even those even though these, this isn't capturing all of the genes by any means, and it isn't capturing by any means all of the somatostatin in that gene, is still enough to identify the cells down to fine subtypes. So to do the identification, we use a probabilistic method. It's a variational Bayesian approach that takes these genes, it models a spatial Poisson process based on the single cell RNA sequencing to tell you what the content of the different cell types is, and it gives you a posterior probability for each cell to belong to any one of the cell type clusters defined by single cell RNA sequencing. So most of them are classified with high probability. This one's got 100 posterior probability. This one's got 88 and 12, but they're for two very well related types. So we do a pretty good job of classifying them, at least according to a Bayesian algorithm giving you a high probability. But that's not enough, because you need to know that the model you put in was valid. So we need some external validation. What we did is take another data set from Alan, which is published by Gowans et al., that was patch-seq in vitro data, and our data set, and we have two independent measures of the depth in the cortex you expect for every cell. And you can see they're very well correlated. Each one of these symbols is one of the fine subtypes, and the X and Y axis tell you the depth measured by our measurement, and Gowans et al., which is an entirely different data set. And the fact that it's so well correlated makes you think that we really are figuring out these cell types accurately, as are Gowans et al. And incidentally, the correlation persists even when you look at the fine subtypes, the ones we don't know what they are biologically. We know that we have consistent measures of layer to this other method. So we're confident that this method works for identifying the fine cell subtypes, even beyond the classically defined cell types. All right, the next thing we need to do is align to in vivo imaging. So we use in vivo imaging in a mouse line that expresses mCherry in inhibitory neurons. This turned out to be the key thing, because registering the ex vivo to in vivo data was incredibly hard. Having these red cells as fiducial points is what made it possible. So we do cell detection in vivo with Sweet2P, and the red cells we identify as interneurons from the in vivo image. So we know where the inhibitory neurons are in vivo. Then we cut brain slices, apply the transcriptomics to it, and in particular label the GAD in red here. So these red cells should correspond to those red cells. We do a search algorithm. It needs to be an exhaustive search to map the brain slice to the 3D Z-stack, exhaustive plus a bit of manual tweaking. And that lets us register this ex vivo section to the in vivo volume and line up the transcriptomics with the in vivo imaging. If you zoom in to a region, you can see in red is the in vivo mCherry that tells us where the inhibitory neurons were in vivo. And in green is the GAD1 detections ex vivo. And the fact that they line up tells you that the alignment has worked. So after all of that work, we finally get some data where we can see the activity of the cells that you get in 2 photon, but now we know what the fine molecular subtypes of each one are. All right, so what's the difference between these fine subtypes? Is it a big difference? Is it a small difference? Is it a difference in the sensory responses of these cells, their spontaneous activity, their relation to ongoing state? What is it? So first we look at sensory responses, and the perhaps surprising answer is that the difference between the fine subtypes for sensory responses is minor at best. So if we look at these, this data shows you just grating responses, but we had other stimuli and got coherent results. If you look at the grating responses, this shows you responses, PSDHs, with each row being a different molecular cell type. Whoops. The average, where did it go? The average over cells in that type in time in response to different sized gratings when the mouse is stationary and when the mouse is running. You can see a big difference between stationary and running. We'll get to that later. But if you look at the sensory responses, you see there's not very much difference, at least between these different types of basket cells, between these different types of Martinotti cells, between the different types of neurogliaform cells, etc. There's certainly differences at the top level. So for example, these SNCG neurons, these are a type of inhibitory neuron that inhibits excitatory cells, but has cannabinoid receptors. These ones, for whatever reason, are uniformly inhibited by visual stimuli. I don't think that was known before. whereas most other cell types are excited by visual stimuli. There's some difference in the size tuning, but there's not much difference. To do the statistics is complicated because different experiments by chance, record different subtypes of cells, and there's experiment to experiment variability, and you don't want to fool yourself that you're seeing a difference between cell types when you're seeing a difference between experiments. But we came up with what we call a hierarchical permutation test, and what it does is it shuffles the cell labels, the molecular subtype labels at different levels of the hierarchy, only within an experiment. And that lets us tell when you've got significant differences at each level of the hierarchy of the cell types. And what you can see here is that for the sensory responses, we've got differences between the top level subclasses, basically that the SNCG cells are inhibited, the rest are excited, but we don't have any significant differences at lower levels of the hierarchy. So the question we started with, do we need to know what the fine subtypes are? If we're looking at sensory responses, the answer is no. Thank goodness we don't need to know what all the fine subtypes are, at least for sensory responses. We only need to know the top level of the hierarchy. All right, but what about spontaneous activity? So this now shows a few minutes of data on a very compressed time scale. This is a raster of the excitatory neurons that we have not molecularly identified. Here you see the running speed of the mouse, the pupil size, and here's a measure of the amount of oscillation in the population activity. So the pyramidal cells, you can see at the end they start oscillating, and presumably the mouse is asleep. We don't have an objective measure of that, but we think the mouse is probably going to sleep, or at least getting drowsy at this point. So the population starts oscillating. And here are all the inhibitory neurons that were recorded in this experiment, and they're colored by their subtypes. And you can see that there's some sort of a switch over at around this time. We've got activity in these ones at the bottom here in the beginning, and then activity of the ones at the top at the end. If we zoom in to 3 sections of running, when the mouse is running, stationary, the mouse isn't running, but we don't have this sleep-like oscillation, and then the sleep-like state at the end, you can see this in more detail. These ones at the bottom, which are the SNCG, VIP, and some of the neurogliaform cells, are active during running. And during the stationary state, we've got the parvalbumin cells and the somatostatin cells, which are more active. And the stationary desynchronized state, when the mouse isn't running, but is still alert, is in between the two. So if we do the same statistical method as before, now we see a difference at all levels of the hierarchy. This is telling us about spontaneous correlations, and this one's telling us about the correlation with running. Let's just look at this one. You see that all the top level of the hierarchy, we have a significant difference between cell types. At the level of types, the ones that we know what classical cell type it corresponds to, we see significant difference for nearly all of the cell types. And even at the level of subtypes, we get a significant difference omnibus, as you see here, and specifically in LAM5, and also in Martinotti cells, although it didn't quite hit our significant threshold. All right, so let's try and, okay, 7 minutes. Let's summarize what's going on. So what we see is that the parvalbumin cells prefer the synchronized state. That's the sleep-like state. The SST Martinotti cells, VIP and SNCG cells, prefer a running state. The neurogliaform cells are mixed, and the neurogliaform cells, remember, are the ones that lay on this continuum. So that the different subtypes here aren't really different cell classes, they're just different positions along this continuum. And the difference between these subclasses that you see here actually reflects a continuum. You can see that if we have a measure of position along the continuum here on the x-axis, and we have a measure of state modulation on the y-axis, you can see a continuous variability. The break between the clusters is arbitrary because it's a continuum of cell types, and we've got a correlation all the way along their continuum. And in fact, we can even see it with single genes. So here, NDNF, the gene we saw earlier that's expressed along this continuum, you can see that the state modulation, the amount it prefers running, correlates with the NDNF expression, even within one of these cell classes. And CCK, a gene that goes in the other direction along the continuum, we have a negative correlation. All right, So we've got this complicated dependence on the transcriptomic classes for running speed correlation. Is there any way to simplify this and make it more interpretable? Well, there is, thankfully, and it's really easy. All you do is PCA, principal component analysis, on the gene counts. So we've got all of our cell types, and we take all of them, we forget what cluster it goes into, and we throw it into principal component analysis. We take the first principal component of the transcriptome, and we get a position for each cell type and for each individual cell along that continuum. And if you look at the genes that are weighted along the continuum, you see the negatively weighted genes are things involved in GABA synthesis, fast spiking, and metabolism. These are cells that are very fast active. And the ones that are positively weighted are genes that are signatures of peptide release, axons in layer 1, that's renin, and excitatory serotonin receptors. If we correlate it again to the Gowans data, you can see that this first transcriptomic principal component analysis correlates with all sorts of in vitro properties of the cells. The subclasses that fire more in the sleep-like synchronized state have larger values of this transcriptomic principal component. They have lower input resistance, faster time constant. They're more hyperpolarized at rest. They need more current to make them fire. They're all adapted to firing rapidly and exerting rapid, strong inhibition on their targets. And they also don't adapt in frequency, so they're giving you strong, powerful, sustained inhibition. The other difference that we see now from the Tasich data that correlates with the state modulation is cholinergic receptor expression. So it's interesting that the ones that are positively state modulated, the ones that fire more in running state and waking state compared to sleep, are the ones that have the excitatory cholinergic receptors. So this is GQ-coupled excitatory muscarinic receptor. These two are nicotinic receptors, and you see they're more expressed in the cells that fire more in the wake-like state, whereas the inhibitory cholinergic receptors are more expressed in the cells that fire in the sleep-like state. And the other difference between them that we saw in the Gowans data is to do with where the axon of these cells are. So to summarize what we see with this transcriptomic axis, it seems like a lot of the properties of all these different cell types, at least to some approximation, can be explained by one single axis of variation, which at least simplifies the data a bit. So the ones with a high transcriptomic principal component, the interneurotypes with a high principal component, are more active in alert states, such as running. They are slower in their time constants. They have adapting spike trains. They're excited by acetylcholine, and they have a lot of axons in layer 1. The other ones at the other end of the continuum, active in the sleep-like states, are faster, and they have more inhibition in the deeper layers. And this is actually coherent with a theory that's been around quite a long time, proposed by Hasselmo and others, which is that what the brain state, changes in brain state do is switch the cortex from a mode where it's driven by bottom-up sensory inputs to a mode where it's driven by top-down feedback. So in a sleep-like mode, in an in-alert mode, the neurons that are active are the ones that are inhibiting let me get this right. They're inhibiting this layer. So the inputs that are spared are the ones coming into layer 1, presumably the top-down inputs. But in an alert mode, when the mouse is engaged behaviorally, it's running, it's these inputs that are suppressed, and the bottom-up inputs from the thalamus to the deeper layers perhaps can persist. So if I've got a couple of minutes left, I'd like to tell you just a bit about, that was all published, a bit about what's going on at the moment. This work takes a long time, but we're starting to get a bit further with it. So the first question is, what about beyond the first principal component? There's this one dimension of transcriptomic activity that correlates very well with the first principal component of the activity of the cells in vivo. What about beyond that single dimension? Of course, there's more going on, and it seems like what we're seeing is a second mode of excitatory activity in vivo correlates with a different transcriptomic axis, which is specifically between somatostatin and SNCG interneurons. So if we look here, this is a piece of data where you're seeing all the different behavioral traces at the bottom. But here we have the activity of the somatostatin interneurons. And you can see they're firing in these very discrete and rare bursts, as you see in the average here. This is the second principal component of activity of the pyramidal cells, correlates very well with somatostatin activity. And the different neurons, different cell types, have a different axis that correlates with it. And it's particularly the SNCG neurons that correlate negatively with it. This second principal component of activity has something to do with saccades. When the mouse makes an eye movement, we see a big increase in the SST Martinotti cells and a big suppression of the SNCG cells. It's not the only time we have this dimension of activity going on, but there's some sort of a correlation. So there's some sort of event that's going on in the cortex that involves the SST bursting, the SNCG suppressed. It happens at the time of saccades, but it also happens at other times too. We're still trying to figure out what that all means. The next thing we want to do, and if I was giving this talk 2 weeks later, I think I'd have some data to present on this, is transcriptomic identification of excitatory cells. And the final thing I want to tell you about is the newest version of the transcriptomic method, Copperfish 3D, which now gives you gene expression data in 3 dimensions, and you can have a great time zooming in. You can just spend hours on looking at these sort of movies. Even if you do it on the London Underground, it's the only time a stranger will come up and talk to you. So what on earth is this stuff? So here you see zooming into different cells of different cell types, and you can swim through it. It's amazing fun. I should say also this method that we developed for in situ transcriptomics, it was based on an earlier collaboration with Mats Nelson from Stockholm, and you can now get commercial systems that do this, but this one is open source, which means that it's substantially cheaper. You can buy a rig and the probes for fairly reasonable price, and if you're interested, please contact me because we'll help you set it up. And with that, I think I'm done. And thanks to the huge team who spent so much time, and to you for your attention. Thanks a lot, Thomas. Thank you.